Go.
So we just came over to the sink and we have a little tub of bleach water in there, and we're gonna empty our Supernate into that bleach to just sterilize anything that might not have pelleted. So we can take one more look at our culture. Again, we have our pellets and our Supernate, which is clear now. So what I just do is I kind of do up and down. So one more time in slow-mo, it was like up and then down, and that's so that I try to minimize the amount of contamination I get. And then I like my pellets really dry, so I just grab a paper towel and slam that down. And these pellets will hold their own, so we can see that they kept their place and that you have a all that extra liquid there. So we're gonna leave all this. We'll clean it up later. But now let's finish our mini prep. So we're gonna come over to my bench. And now we're going to add our P1. So from here on, all the volumes are going to be same as a normal mini prep. And the P1 comes out of the fridge. I already took it out, but it has some DNase in it, or I'm sorry, RNase A, and that will slowly degrade at room temperature. So that always needs to be kept in the fridge when you're not using it. So as the regular mini prep protocol says, 250 microliters of P1 per sample. So all we're going to do is 250. I'm just going to go all the way through this. And meanwhile, you always want to be checking your pipette tip, making sure that you have about the same volume each time. And again, I'm using good pipette technique here. I'm not lifting up my thumb on the plunger too quick. I'm just kind of doing a gradual lift up. Otherwise you'll get air bubbles. If I lift it up too quick, you'll get, well, didn't work that time. Here, I'll show you. This is what happens if you lift it up too quick. That's no good. So I'm checking to make sure I don't get any of that. So I've put P1 in all of my wells. I'm now going to cap the P1 and put it back in the fridge. So the refrigerator, not the freezer. So we'll just put it right here for now. Now we're going to resuspend it. So the P1 resuspends our pellets in the conditions that will allow the rest of the mini prep to work properly. So I just get a new strip. This is not an air pour strip. This is I think an air seal. I forgot what it's called. Seal plate strip. It's kind of like sticky on one side, and you can just close it on the other. And then I come over to the vortex, and I just hit the vortex for a little while and let it resuspend. So I'm going to be looking at it every now and again. If I kind of hold it at an angle, we can see that the pellets have mostly disappeared. We can also hold it up to the light, and the pellets have all disappeared, so we've resuspended everything. I'm just going to buzz it one more time for good luck. Tap it down. Get any liquid that is splashed up. So you can see that on the top we have a little bit of liquid here. So this is a goner that can lead to contamination. So I just fold that in half and toss that. And now we're going to go into buffer 2, P2, which is our lysis. So now all of our bacteria has been resuspended in our P1. It's in conditions where the rest of the buffers will have the right type of activity. So I'm running a little bit low on P2, so I need to be extra careful with my pipette here. So 250 microliters of P2 to each sample. This is the lysis buffer. It's just dissolving all of the cells. It's cracking them open. The cells are now releasing the contents into the solution, including their plasmids. And you always want to be keeping extra careful track of where you are in terms of your liquid handling, because it's easy to get mixed up and lose track and maybe skip your P2 for a sample or double dose on P2 for a sample, which again will just mess up all of your experiments, so don't do that. So just 3 more left. Checking a pipette each time before you put the liquid in to make sure that you're putting that proper volume in. So that's all of them. So P2 is not temperature sensitive, so this can just stay right up here. And then we want to be very gentle during this lysis step. We're going to rock it very slowly just for a little while, being very gentle. If we rock it too hard, like if I took my block and I started rocking it like that, then I would be shearing the genome. And if we shear the genome, that means we're going to get fragments that are about plasmid size, so they'll behave more like a plasmid, so that when we proceed with the rest of our mini prep, it'll get purified along with our plasmid, contaminating it. So instead of having a sample with just plasmid DNA, we're going to get a sample with plasmid DNA and a bunch of fragments of our genomic DNA, which is just, it gives us a smear. It's just bad news. It's just contaminant. And we want to be gentle because if we do fragment, if we do shear that genome, it's not going to be able to get crashed out in the next step that we're about to do, which is a neutralization step. So take a look at your culture. It's now clear. It's no longer cloudy like it was. That P2 did its magic, busted everything open, and now we're going to get rid of all of the cell walls, all of the membranes, all the proteins, and all of the genomic DNA, and I'm adding in 350 microliters of N3. So that's neutralization buffer. And at this point, we can just kind of go over the buffers. There's 3 buffers, P1, P2, N3. So 1, 2, 3. P1 and P2 are 250. N3 is 350 microliters. So double checking the volumes. And you just want to remember that protocol. So 1, 2, 3, P1, P2, P3. And that's really all there is to a mini prep. And just keeping track of when you shake vigorously versus when you kind of rock, and you want to pay attention so that you don't miss any of your wells or do anything that might mess up your mini prep, because once you get this down, it's a pretty easy procedure, but you need to do it the right way. If you learn it the wrong way, then you can really mess things up. If you skip a step or if you do something systematic, you can really do a lot of long-term damage to your science. For a long time, I was missing a wash step, which we'll get to in a little while, which really messed me up. It degraded all my DNA. So you really want to make sure that you do this right. This is one of the most fundamental techniques in microbial biology or in synthetic biology. So we have N3 in all of our wells. Now we can look at it, we can mix it up a little bit. I'm actually going to get a new one of those plate seals. And we want to be a little bit more vigorous when we start to mix this up. But since we still have a lot of that liquid in the P2 state, we have to start slowly, and then we can work our way up. So what happens when we add the N3 is it starts to crash out a lot of the junk that's in our lysate, and it will create a film. So we have, if you can imagine, an oil and water. If, in this example, our P1, P2 mixture is like the water, and the N3 is like the oil. So we have kind of an interface, and when the N3 mixes with that P2, it creates a membrane of stuff that gets crashed out, which means that the N3 cannot diffuse through that membrane, which means we need to rip up that membrane to make sure that the P2 and the N3 keep on mixing with each other. So I get more and more vigorous, and I'm kind of checking it each time. And what I want to see is, I want to see a lot of little mini chunks. Because what happens is, in this example, in this well, if you can see, there's one big chunk. But if we look at the well right next to it, right there, there's a bunch of small chunks. That's what we want to see. We want to make sure that all of our wells like that, because that means that there's no pockets of N3 that haven't had contact with the rest of the solution, and that we've really crashed everything out of our mini prep mix. So double checking it, and you got to get vigorous and you got to shake it for a little while. But if you don't really break that pellet up, you're going to have a bad mini prep. So a little bit more. Looks like we're getting good right now. The middle one is not cooperating though. Okay. So I think we're good. Now we're going to spin this down. We're going to head to the centrifuge real quick, and we're going to do the same thing that we did before. So we're going to weigh our block. Block should be a little bit lighter now, so 189.2. I'm going to pour off some of my water here. And let's see how close we are. So 180.2, so 9 away. So again, I'm just filling water up around the outside. .82, 1 more gram. 189.3, that's close enough. And then we're just going to put our blocks into the rotor. Close the rotor. Always double check your rotor. S5700, that's good. RPM 5400. Instead of 3 minutes, we're going to be spinning for 10 minutes. Enter. Start. And we'll be back in about 10 minutes. So